That dream about the dinosaur and the leotard, and those times that you said that thing that you know you shouldn't have said, or even that thing you didn't even know you were gonna say. The little cogs of your consciousness cranking away, making your life possible, making society function, all of the things that you're so glad you can do, and all the ones you wish you could stop doing. Excluding other human minds, your mind is the most complicated piece of the universe that humans currently know about. The rules that govern it are mysterious and elusive. Maybe our brains just aren't complex enough to understand themselves, but that's not going to stop us from trying. The word psychology comes from the Latin for the study of the soul, and while its formal definition has evolved over the last several decades, today we can safely call it the science of behavior and mental processes. The term psychology wasn't coined until around the turn of the 16th century, and the practice that we would actually call science today wasn't established until the mid-1800s. But of course, humans have always been curious about themselves and what's going on up here. Aristotle pondered the seed of human consciousness and decided that it was in the heart, not the head, being, as we have seen quite a lot here in Crash Course, absolutely and completely wrong. 2,000 years ago, Chinese rulers conducted the world's first psychological exams requiring public officials to take personality and intelligence tests. And in the late 800s, Persian Dr. Muhammad ibn Zakaria al-Razi, also known as Razis, was one of the first to describe mental illness and even treated patients in what was essentially a very early psych ward in the Baghdad hospital. From the efforts of those early thinkers up until today, the field of psychology has been all about tackling some of the big questions. How can humans do horrible things like commit genocide and torture other humans, and how come we know those things are horrible? Do we have free will, or are we simply driven by our environment, biology, and non-conscious influences? What is mental illness, and what can we do about it? And what is consciousness, or the notion of self? If I lose my awareness of myself, am I still human? I don't know. But over the next 6 months, these are the questions that we're going to be exploring together. How our brains work, how they can break, how they can be healed, why we behave the way we do, even when we don't want to, and what it means to be thinking, feeling, and alive. When hearing the word psychology, most people probably think of a therapist listening to a patient unpacking the details of his day while they're planning on a couch. Maybe that therapist is wearing glasses, chewing on a cigar, stroking his whiskered chin. Admit it, if you're thinking about psychology, you're probably picturing Freud. Sigmund Freud was one of the most tremendously influential and controversial thinkers of his time, maybe of all time. His theories helped build our views on childhood, personality, dreams, and sexuality, and his work fueled a legacy of both support and opposition. His life was long and spanned an important swath of history from the American Civil War to World War II. But like most great scientists, Freud developed his revolutionary ideas by building on the work of others. And of course, innovation in the field didn't stop with him. In truth, psychology is one of the most wildly diverse sciences in terms of the questions it poses, the methods it applies, and the different schools of thought and the disciplines it contains. Perhaps more than any other science, psychology is just a big old integrated melting pot. For instance, right around Freud's time, there were a lot of different schools of thought about how the study of the human mind should be tackled. Mainly, there were the ideas of structuralism, functionalism, and psychoanalysis. Scientific psychology got its start in 1879 in Germany when physician Wilhelm Wundt set up the first psychology laboratory at the University of Leipzig just a few years after publishing his Principles of Physiological Psychology, considered the first true psychology textbook. Wundt and his student Edward Bradford Titchener took cues from chemists and physicists and argued that if those people could break down all matter into simple elements or structures, why couldn't they do the same for the brain? They tried to understand the structures of consciousness by getting patients to look inward, asking them how they felt when they watched the sunset, or smelled coffee, or licked the kitten, or whatever. Titchener named this approach structuralism, but despite its rigid-sounding name, it really relied so much on introspection that it became too subjective. I mean, you make sense and feel something different than I do, even if we lick the same kitten. Psychologists, of course, can't actually observe a patient's inner thoughts or feelings, so ultimately the structuralist school of thought is fairly short-lived. By contrast, American physician and philosopher William James proposed a different set of questions, focusing on why we think and feel and smell and lick, whatever. Basically, he focused on the function of behavior. This approach, functionalism, was inspired by Charles Darwin's idea that adaptive behaviors are conserved throughout the evolutionary process. James published his seminal book, The Principles of Psychology, in 1890, defining psychology as the science of mental life. Just as Freud was starting to flex his big brain. Sigmund Freud began his medical career at a Viennese hospital, but in 1886, he started his own practice specializing in nervous disorders. During this time, Freud witnessed his colleague Joseph Breuer treat a patient called Anna O with a new talking cure. Basically, it just let her talk about her symptoms. The more she talked and pulled up traumatic memories, the more her symptoms were reduced. It was a breakthrough, and it changed Freud forever. From then on, Freud encouraged his patients to talk freely about whatever came to mind, to free associate. This technique provided the basis for his career and an entire branch of psychology. In 1900, he published his book, The Interpretation of Dreams, where he introduced his theory of psychoanalysis. Now, you probably think of psychoanalysis as a treatment, the whole patient on the couch scenario, and that's definitely part of it. But Freud's concept was actually a lot more complex than that, and it was revolutionary. The radical kernel of psychoanalysis was the theory that our personalities are shaped by unconscious motives. Basically, Freud suggested that we're all profoundly affected by mental processes that we're not even aware of. Now that sounds almost obvious to us now, but part of the genius of Freud's theory was that in 1900, it wasn't obvious at all. The idea that our minds could be driven by something that our minds themselves didn't know about was hard to grasp. As hard as, maybe organisms evolving by natural selection. It was abstract, invisible, and there was something about it that seemed irrational. But the other important part of Freud's theory was that the subconscious, literally the thing below consciousness, was still discoverable. Even though you weren't aware of it, you could come to understand it through a therapeutic technique that used dreams, projections, and free association to root out repressed feelings and gain self-insight. So what Freud was really saying was that mental disorders could be healed through talk therapy and self-discovery. And this was a really big breakthrough, because prior to this, people with mental illnesses would be confined to sanatoriums and at best given menial labor to do, and at worst, shackled to a bed frame. After the interpretation of dreams, Freud went on to publish over 20 more books and countless papers with an iconic cigar in hand all the while. He believed smoking helped him think, but also helped him get jaw cancer. During the last 16 years of his life, he underwent at least 30 painful operations while continuing to smoke. By the late 1930s, the Nazis had taken over Austria, and Freud and his Jewish family narrowly escaped to England. By September 1939, the pain in his cancerous jaw was too great, and a doctor friend assisted him in suicide through morphine injection. He was 83. Whether you love him or hate him, and make no mistake, plenty of people vehemently disagreed with him, there is no question that Freud's impact on psychology was monumental. While competing theories in the young field of psychology either fell away or evolved into something else, psychoanalysis remains an important concept and practice today. The next big shakeup rolled in during the first half of the 20th century when behaviorism gained a higher profile. Heavy hitters like Ivan Pavlov, John B. Watson, and B.F. Skinner were key players here. They focused on the study of observable behavior. You may remember Skinner as the dude who put rats and pigeons and babies in boxes and conditioned them to perform certain behaviors. Right around when Freud escaped to England, Skinner published his Behavior of Organisms, ushering in the era of behaviorism, which remained all the rage well into the 1960s. The other major force at the time was, of course, Freud's psychoanalysis and its many descendants, collectively known as the Psychodynamic Theories. These focused on the importance of early experiences in shaping the unconsciousness and how that process affects our thoughts, feelings, behaviors, and personalities. By the mid-20th century, other major forces in psychology were also brewing. Schools we'll explore later in this course, including humanist psychology, which focuses on nurturing personal growth, cognitive science, and neuroscience, all of which contributed their own unique takes on the study of mind. Today's formal definition of psychology, the study of behavior and mental processes, is a nice amalgamation that pulls from all these different schools of thought. It recognizes the need for observing and recording behavior, whether that's screaming, crying, or playing air saxophone to an imaginary audience, but it also gives credit to our mental processes, what we think, feel, and believe while we're tearing it up on our invisible instruments. Because again, the point that I really want you to take home is that psychology is an integrated science. Yes, folks will get grumpy and disagree plenty, but the essence of the discipline has everything to do with creating different ways of asking interesting questions and attempting to answer them through all kinds of data gathering methods. The human mind is complicated. There is no single way to effectively crack it open. It must be pried at from all sides. Harvard astronomer Owen Gingerich has gazed into the distant horizons of space, and even he has acknowledged that the human brain is by far the most complex physical object known to us in the entire cosmos. And we all get to have one. of our very own. Knocking around right up in here. We here at Crash Course are really excited to spend the next several months delving into the world of psychology, how it applies to our lives, our minds, and our hearts, and how it deepens our understanding of each other, our world, and ourselves. Thanks for watching this first lesson in Crash Course Psychology, and I'd like to especially thank all of our subbable subscribers, without whom we would literally not be able to do this. Would you like a personalized signed Crash Course Chemistry periodic table, or even to see yourself animated in one of our episodes? To find out about these and other perks, go to subbable.com/crashcourse. And thanks to our crew. This episode was written by Kathleen Yale and edited by Blake T. Pastino. Our psychology consultant is Dr. Ranjit Bhagwat. Our director and editor is Nicholas Jenkins. The script supervisor was Michael Randa, who is also our sound designer. And our graphics team is Don Cafe. Can we go pizza called psychedelic hallucinations? Does coffee make you smarter? Or does it just make you do dumb stuff faster? Like much of psychology itself, questions like these can seem pretty intuitive. I mean, people may not be the easiest organisms to understand, but you're a person, right? So you must be qualified to draw like some conclusions about other people and what makes them tick. But it's important to realize that your intuition isn't always right. In fact, sometimes it is exactly wrong, and we tend to grossly underestimate the dangers of false intuition. If you have some idea about a person and their behavior that turns out to be right, that reinforces your trust in your intuition. Like if I warn my buddy Bob against eating that deep dish pizza that's been in the fridge for the past week, but he eats it anyway and soon starts to wig out, I'm gonna say, dude, I told you so. But if I'm wrong and he's totally fine, I probably won't even think about it ever again. This is known as hindsight bias, or the I knew it all along phenomenon. This doesn't mean that common sense is wrong, it just means that our intuitive sense more easily describes what just happened than what will happen in the future. Another reason you can't blindly trust your intuition is your natural tendency toward overconfidence. Sometimes you just really feel like you're right about people when actually you're really wrong. We've all been there. We also tend to perceive order in random events, which can lead to false assumptions. For example, if you flip a coin 5 times, you have equal chances of getting all tails as you do getting alternating heads and tails. But we see the series of 5 tails as something unusual, as extreme, thus giving that result some kind of meaning that it very definitely does not have. That is why we have the methods and safeguards of psychological research and experimentation, and the glorious process of scientific inquiry. They help us to get around these problems and basically save the study of our minds from the stupidity of our minds. So I hope that it won't be a spoiler if I tell you now that pizza won't make you trip, and coffee doesn't make you smart. Sorry. In most ways, psychological research is no different than any other scientific discipline. Like, step 1 is always figuring out how to ask general questions about your subject and turn them into measurable, testable propositions. This is called operationalizing your questions. So you know how the scientific method works. It starts with a question and a theory. And I don't mean theory in the sense of like a hunch that, say, a quad shot of espresso makes you think better. Instead, in science, a theory is what explains and organizes lots of different observations and predicts outcomes. And when you come up with a testable prediction, that's your hypothesis. Once your theory and hypothesis are in place, you need a clear and common language to report them with. So, for example, defining exactly what you mean by thinking better with your espresso hypothesis will allow other researchers to replicate the experiment. And replication is key. You can watch a person exhibit a certain behavior once, and it won't prove very much. But if you keep getting consistent results, even as you change subjects or situations, you're probably onto something. This is a problem with one popular type of psychological research, case studies, which take an in-depth look at one individual. Case studies can sometimes be misleading because by their nature, they can't be replicated, so they run the risk of overgeneralizing. Still, they're good at showing us what can happen and end up framing questions for more extensive and generalizable studies. They're also often memorable and a great storytelling device psychologists use to observe and describe behavior. Like, say the smell of coffee makes Carl suddenly anxious and irritable. That obviously doesn't mean that it has that same effect on everyone. In fact, Carl has terrible memories associated with that smell, and so his case is actually quite rare. Poor Carl. But you would still have to look at lots of other cases to determine that conclusively. Another popular method of psychological research is naturalistic observation, where researchers simply watch behavior in a natural environment. Whether that's chimps poking anthills in the jungle, kids clowning in a classroom, or drunk dudes yelling at soccer games. The idea is to let the subjects just do their thing without trying to manipulate or control the situation. So yeah, basically just spying on people. Like case studies, naturalistic observations are great at describing behavior, but they're very limited in explaining it. Psychologists can also collect behavioral data using surveys or interviews, asking people to report their opinions and behaviors. Sexuality researcher Alfred Kinsey famously used this technique when he surveyed thousands of men and women on their sexual history and published his findings in a pair of revolutionary tests, Sexual Behavior in the Human Male and Female, respectively. Surveys are a great way to access people's consciously held attitudes and beliefs, but how to ask the questions can be tricky. Subtle word choices can influence results. For example, more forceful words like ban or censor may elicit different reactions than limit or not allow. Asking, do you believe in space aliens, is a much different question than, do you think that there is intelligent life somewhere else in the universe? It's the same question, but in the first, the subject might assume that you mean aliens visiting Earth and making crop circles and abducting people and poking them. And if how you phrase surveys is important, so is who you ask. I could ask a room full of students at a pacifist club meeting what they think about arms control, but the results wouldn't be a representative measure of where students stand because there's a pretty clear sampling bias at work here. To fairly represent the population, I'd need to get a random sample where all members of the target group, in this case students, had an equal chance of being selected to answer the question. So once you've described behavior with surveys, case studies, or naturalistic observation, you can start making sense out of it and even predict future behavior. One way to do that is to look at how one trait or behavior is related to another, or how they correlate. So let's get back to my buddy Bob, who seems to think that his refrigerator is actually some kind of time machine that can preserve food indefinitely. Let's say that Bob is just tucked into a lunch of questionable leftovers, pizza that may very well have had a little bit of fungus on it, but he was hungry and lazy, and so he doused it in sriracha. Suddenly, he starts seeing things. Green armadillos with laser beam eyes. From here, we could deduce that eating unknown fungus predicts hallucination. That's a correlation. But correlation is not causation. Yes, it makes sense that eating questionable fungus would cause hallucinations, but it's possible that Bob was already on the verge of a psychotic episode, and those fuzzy leftovers were actually benign. Or there could be an entirely different factor involved, like maybe he hadn't slept in 72 hours or had an intense migraine coming on, and one of those factors causes hallucinations. It's tempting to draw conclusions from correlations, but it's super important to remember that correlations predict the possibility of cause and effect relationships. They cannot prove them. So we talked about how to describe behavior without manipulating it and how to make connections and predictions from those findings, but that can only take you so far. To really get to the bottom of cause and effect behaviors, you're gonna have to start experimenting. Experiments allow investigators to isolate different effects by manipulating an independent variable and keeping all other variables constant, or as constant as you can. This means that they need at least two groups: the experimental group, which is gonna get messed with, and the control group, which is not gonna get messed with. Just as surveys use random samples, experimental researchers need to randomly assign participants to each group to minimize potential confounding variables or outside factors that may skew the results. You don't want all grubby teenagers in one group and all wealthy Japanese servers in the other. You gotta mingle. Now, sometimes one or both groups are not informed about what's actually being tested. For example, researchers can test how substances affect people by comparing their effects to placebo or inert substances. And often, the researchers themselves don't know which group is experimental and which is control, so they don't unintentionally influence the results through their own behavior, in which case it's called, you guessed it, a double-blind procedure. So let's put these ideas into practice in our own little experiment. Like all good work, it starts with a question. So the other day, my friend Bernice and I were debating. We're debating caffeine's effect on the brain. Personally, she's convinced that coffee helps her focus and think better, but I get all jittery like a caged meerkat and can't focus on anything. And because we know that overconfidence can lead you to believe things that are not true, we decided to do some critical thinking. So let's figure out our question. Do humans solve problems faster when given caffeine? Now we gotta boil that down into a testable prediction. Remember, keep it clear, simple, and eloquent so that it can be replicated. Caffeine makes me smarter is not a great hypothesis. A better one would be, say, adult humans given caffeine will navigate a maze faster than beings not given caffeine. The caffeine dosage is your independent variable, the thing that you can change. So, what is in common? Your result or dependent variable, the thing that depends on the thing that you can change, is going to be the speed at which the subject navigates this giant corn maze. Go out on the street, wrangle up a bunch of different kinds of people, and randomly assign them into 3 different groups. Also, at this point, the American Psychological Association suggests that you acquire everyone's informed consent to participate You don't want to force anyone to be in your experiment, no matter how cool you think it is. So the control group gets placebo, in this case decaf. Experimental group 1 gets a low dose of caffeine, which we'll define at 100 milligrams, just an eye-opener and like a cup of coffee's worth. Experimental group 2 gets 500 milligrams, more than a quad shot of espresso dunked in a Red Bull. Once you dose everyone, turn them loose in the maze and wait at the other end with a stopwatch. All that's left is to measure your results from the 3 different groups and compare them to see if there were any conclusive results. If the highly dosed folks got through it twice as fast as the low dose placebo groups, then Bernice's hypothesis was correct and she could rub my face in it saying she was right all along. But really, that would just be the warm flush of hindsight bias telling her something she didn't really know until we tested it. Then, because we've used clear language and defined our parameters, other curious minds can easily replicate this experiment and we can eventually pull all the data together and have something solid to say about what that macchiato was doing to your cognition, or at least the speed at which you can run through a maze. Science, probably the best tool that you have for understanding other people. Thanks for watching this episode of Crash Course Psychology. If you paid attention, you learned how to apply the scientific method to psychological research through case studies, naturalistic observation, surveys, and interviews and experimentation. You also learned about different kinds of bias and experimentation and how research practices help us avoid them. Thanks especially to our subbable subscribers who make this and all of Crash Course possible. If you'd like to contribute to help us keep Crash Course going and also get awesome perks like an autographed science poster or even be animated into an upcoming episode, go to subbable.com/crashcourse to find out how. Our script was written by Kathleen Yale and edited by Blake Hughes and myself. Our consultant is Dr. Ranjit Bhagwan. Our director and editor is Nicholas Jenkins. Our script supervisor is Nicola Rhonda, who is also our sound designer and our graphics team. It's Thought Cafe. It's late at night. You're home alone, drifting off to sleep, entering that dream about freedoms, and then suddenly there's a banging at the door. Suddenly, you're wide awake, and it feels like your heart's gonna explode. You jump up, ready to run out the back door, possibly grab a Phillips head screwdriver and stab it into the darkness until it sticks into something. Now, whether it's a weeping angel or your neighbor looking to borrow a can of beans, doesn't really matter, because when you heard that sudden noise, your startled brain released an icy typhoon of chemicals, and everything that's now going through your mind, like your urge to flee, your urge to defend yourself, that internal debate about whether weeping angels are even real, and, well, where's the cat? All that is just the result of those chemicals. Our brains and our nervous systems and the substances they produce and are always bathed in are amazingly complex, nuanced systems. And even though we're always talking about our mental activity as being somehow separate from all the biological stuff going on in our bodies, in reality, the moods, ideas, impulses that flash through our minds are spurred by our biological condition. As psychologists like to say, everything psychological is biological. So one way to understand how your mind works is to look at how the chemistry of your body influences how you think, sense, and feel about the world around you. To do that, we begin at the simplest level, the system with the smallest parts. It's all about the neuron, baby. Neurons, or nerve cells, are the building blocks that comprise our nervous systems. Neurons share the same basic makeup of our other cells, but they have electrochemical mojo that lets them transmit messages to each other. Your brain alone is made up of billions of neurons, and to understand why we think or dream or do anything, you gotta first understand how these little transmitters work. Actually, there's several different types of neurons in your body, from ones that are less than a millimeter long in your brain to ones that run the whole length of your leg. Yes, you have cells as long as your legs, which is nothing compared to the 150 feet the nerve cells of some dinosaurs had to be. I'm getting off topic, sorry. No matter how big a nerve is, they all have the same 3 basic parts: the soma, dendrites, and axon. The soma, or cell body, is basically the neuron's life support. It contains all that necessary cell action like the nucleus, DNA, mitochondria, ribosomes, and such. So if the soma dies, the whole neuron goes with it. The dendrites, as bushy and branch-like as the trees they're named after, receive messages and gossip from other cells. They're the listeners whispering what they hear back to the soma. The axon is the talker. This long cable-like extension transmits electrical impulses from the cell body out to other neurons, or glands, or muscles. Whereas the dendrites are short and bushy, the axon fiber is long, and depending on what type of neuron it is sometimes encased in a protective layer of fatty tissue called the myelin sheath. It's almost like an insulated electrical wire. The myelin sheath speeds up the transmissions of messages, and if it degrades, as it does with those affected with multiple sclerosis, those signals are degraded as well, eventually leading to lack of muscle control. Neurons transmit signals either when stimulated by sensory input or triggered by neighboring neurons. The dendrites pick up the signal and activate the neuron's action potential or firing impulse that shoots an electrical charge down the axon to its terminals and toward the neighboring neurons. The contact points between neurons are called synapses. All those bushy little dendrites are decorated with synapses that almost, but don't quite touch the neighboring axon. The tiniest game of "I'm not touching you" of all time. They're less than a millionth of an inch apart, and that microscopic cleft is called the synaptic gap. So when an action potential runs down to the end of an axon, it activates the chemical messengers that jump that tiny synaptic gap, flying like that little air kiss, landing on the receptor sites of the receiving neuron. Those messengers are neurotransmitters. Although neurotransmitters slide right into their intended receptors like a key into a lock, they don't stay bonded to the receiving neuron. They just sort of pop out, having excited or inhibited the receiving neuron's trigger, then the extras immediately get reabsorbed by the neuron that released them in the first place, in a process called reuptake. Kind of like, here you go, bye. So neurons communicate with neurotransmitters, which in turn cause motion and emotion. They help us move around, make jazz hands, learn, feel, remember, stay alert, get sleepy, and pretty much do everything we do. Some of them just make you feel good, like the endorphins we get flooded with after running 10 miles or falling in love or eating a really good piece of pie. We've got over 100 different kinds of these brilliant neurotransmitters. Some are excitatory and others are inhibitory, and all are good reminders that everything psychological is also biological. Excitatory neurotransmitters rev up the neuron, increasing the chances it will fire off an action potential. Norepinephrine is one you're probably familiar with that helps control alertness and arousal. Glutamate is another involved in memory, but an oversupply of it can wig out the brain and cause seizures and migraines, which is why some people are sensitive to all that MSG or monosodium glutamate in their ramen. Inhibitory neurotransmitters, on the other hand, chill neurons out, decreasing the likelihood that the neuron will jump into action. GABA, gamma-aminobutyric acid, is a major inhibitory neurotransmitter, and you've probably heard of serotonin, which affects your mood and hunger and sleep. Low amounts of serotonin are linked to depression, and certain classes of antidepressants help raise serotonin levels in the brain. Some neurotransmitters, like acetylcholine and dopamine, play both sides and can both excite or inhibit neurons, depending on what type of receptors they encounter. Acetylcholine enables muscle action and influences learning and memory. Alzheimer's patients experience a deterioration of the acetylcholine-producing neurons. Dopamine, meanwhile, is associated with learning, movement, and pleasurable emotions, and excessive amounts of it are linked to schizophrenia as well as addictive and impulsive behaviors. Some neurotransmitters are basically your nervous system's couriers, but they aren't the only chemical messengers delivering the news. They've got some competition brewing in the endocrine system. Have you been through puberty? You know what I'm talking about. Hormones. Like neurotransmitters, hormones act on the brain, and indeed, some of them are chemically identical to certain neurotransmitters. Hormones affect our moods, arousal, and circadian rhythm. They regulate our metabolism, monitor our immune system, signal growth, and help with sexual reproduction. You could say that most of them boil down to the basics: attraction, appetite, and aggression. Whereas neurons and synapses flick on and off, sending messages with amazing speed, the endocrine system likes to take its time, delivering the body's slow chemical communication through a set of glands that secrete hormones into the bloodstream, where they're ferried to other tissues, especially the brain. So while the nervous and endocrine systems are similar in that they both produce chemicals destined to hit up certain receptors, they operate at very different speeds. It's like if the nervous system wants to get in touch with you, it sends you a text. But if the endocrine system has a message, it will lick the stamp and put it on, write your address, and then the note, and the pen on paper, and then fold it up and put and mail it to you with the post office. But fast isn't always better, and your body will remember that letter longer than the text. Hormones they linger, which helps explain why it takes some time to simmer down after a moment of severe fright or anger. And our endocrine systems have a few important hormone-brewing glands. We've got a pair of adrenal glands snuggled up against their kidneys that secrete adrenaline, that famous fight-or-flight hormone that jacks up your heart rate, blood pressure, and blood sugar, giving you that tidal wave of energy, preparing you to run like heck or punch that charging baboon in the throat. The pancreas sits right next to the adrenal gland and oozes insulin and glucagon hormones that monitor how you absorb sugar, your body's main source of fuel. Your thyroid and parathyroid glands at the base of your throat secrete hormones that regulate your metabolism and monitor your body's calcium levels. If you have testicles, they're secreting your sex hormones like estrogen and testosterone. And if you got ovaries, they're doing that job. And all those glands are super important, but there is one gland that rules them all, and the darkness binds them. The pituitary. Although it's just a little pea-sized nugget hidden deep in the bunker of the brain, it is the most influential gland in this system. It releases a vital growth hormone that spurs physical development, and that love hormone, oxytocin, that promotes warm and fuzzy feelings of trust and social bonding. What really makes the pituitary the master gland is that its secretions boss around the other endocrine glands. But even the pituitary has a master in the hypothalamus region of the brain, which we will talk more about next episode. So, if I managed to scare you, sorry, but I'm illustrating a point. You have no control over being scared, but maybe now you do understand a little more clearly how your nervous and endocrine systems work together to call the shots. First, the sensory input from your eyes and ears went to your brain, the simplest bits of your hypothalamus without even letting you analyze it, and we're like, ah! And then that ran down the chain of command from your pituitary to your adrenal glands, to the hormone adrenaline, to the rest of your body, and then back to your brain, which then realized that I was just messing with you and told everybody to just calm down for once. The whole deal is a feedback loop. Your nervous system directs your endocrine system, which directs your nervous system, brain, gland, hormone, brain. And of course, each of these systems is fantastically complex, way more than we can get into here. So in our next lesson, we're going to get all up in your brain and delve deeper into different components of your nervous system, find out what your old brain is, and learn about how much of your brain you actually use. In the meantime, thank you for watching this lesson in Crash Course Psychology, which was brought to you by Zane Ice, who wants to say hi to his friend Harrison. Thank you, Zane. If you'd like to sponsor an episode and give your own shout out, you can learn about that and other perks available to our Subbable subscribers. Just go to subbable.com/crashcourse. This episode was written by Kathleen Yale, edited by Blake DiGastino, and our consultant is Dr. Ranjit Bhagwan. Our director and editor is Nicholas Jenkins. The script supervisor is Michael Lavanda, who is also our sound designer, and the graphics team is Todd Cafe. In the early 1800s, German physician Franz Joseph Gall spent a lot of time running his fingers over the scalps of strangers. He wasn't a hairdresser, he wasn't a masseuse, he wasn't like just a big fan of heads. He was a phrenologist. He was the first phrenologist. Gall believed that a person's personality was linked to their skull morphology, that its bumps and ridges indicated aspects of their character. Amazingly, this science actually caught on, was widely practiced for decades, and Gall became something of a celebrity. Well, with a head like his, one can see how he might have been a little bit fixated with skull shape. Eventually, phrenology was dismissed as a cult pseudoscience because it turns out that your cranial contours tell us exactly nothing about what's happening inside the brain. And yet, Gall was actually on something big, something that we knew nothing about. Remember, at this point, we were just starting to get consensus that the brain was the source of self, and not like the soul or the heart or whatever. His lasting and correct proposition was that different parts of the brain control specific aspects of our behavior. Like we talked about last time, there is a strong link between biological activity and psychological events. But in addition to the interplay of chemicals like neurotransmitters and hormones, a lot of this also has to do with the fact that localized parts of the brain have specific functions, like vision, movement, memory, speech, even facial recognition. Function, in other words, is localized. If you could stimulate different parts of my brain any way you wanted to, and if you ask me nicely, I just might let you. You can control my movements, my memories, even my personality. Poke my brain over here, and my arm would twitch. Poke it here, and I remember my first kiss. Do it up here, and suddenly I'd be filled with a tremendous Hulk-like rage. This is about the link between the brain, that physical hunk of gunk between the ears, and the mind. The thing that is us, our consciousness, our behavior, our decisions, our memories, ourselves. Some neuroscientists like to say that the mind is what the brain does. So one of the driving questions of psychology and like the human experiment is how do our brains functions tied to the behavior of the mind? And you can't even ask the right questions, let alone get some of the answers, until you get to know the brain. You might have a passing familiarity with your nervous system. Like the brain bone is connected to the spinal cord bone, and spinal cord bone is connected to the motor neuron bone. That's your central nervous system, and there aren't actually any bones. The central nervous system, or just CNS, is what makes your body's big decisions. This system is the command center, and if you mess with it, things are going to get weird. There's also the peripheral nervous system, which is composed of scout-like sensory neurons that gather information and report it back to the central nervous system. But to get a handle on just how physical the roots of your mind and personality are, how concretely your nervous system makes you you, let me tell you a story. The curious case of Phineas Gage. In 1848, a genial chap named Phineas Gage was working on the railroad, tamping gunpowder into a blasting hole with an iron rod, and the gunpowder ignited. The resulting explosion caused the rod to shoot like a bullet up through his left cheek and out of his head. There's brain in between those two places, by the way. Amazingly, he stood up after the accident, walked over to a cart, described what had happened, and then they drove him back to his house, all while he was conscious. So the doctor came to examine him and refused to believe that a rod had in fact passed through his head, understandably, until Phineas started coughing, and an amount of brain that the doctor described as a teacup full fell out of his head. And the doctor had to accept, indeed, what had happened. After a few months of convalescing, he was pretty much healed up and moving around like he used to. But some of his friends were saying that Phineas was no longer himself. Yes, he had his memories and mental abilities, and he walked and talked and looked the same minus an eyeball, but whereas the old Phineas was mild-mannered and soft-spoken, the post-spike brain Phineas was surly, mean-spirited, and vulgar. People started to describe him as no longer Gage. Phineas moved away from America, the scientific establishment lost contact with him, and 12 years later, after a series of seizures, he died at the age of 36. Phineas is a great, if extreme, example of how function is localized in the brain and how physical biological factors can be reflected in psychological ways. Of course, he's also an excellent example of how individual case studies are not particularly useful, especially since we have very little data on what Phineas was actually like before or even after his accident. Most accounts are from the months directly after the accident, and many of them conflict. It's completely possible that he continued to heal and lived his remaining years as a happy and productive citizen. Intro psychology texts often paint a pretty simple picture of Phineas, just so that we can have a clear example of the moment when physicians realized that messing with the brain was messing with the mind, but it is, of course, all much more complicated, and as Phineas was an actual real-life person, I feel like we should give him the nuance and mystery that he deserves. I might have heard that we only use about 10% of our brains, and oh, if that were true, Phineas could lose a quarter of his, and he'd be just fine. And if we could just harness the rest of that gray mush, we'd be able to mind read and levitate and get off Professor X. It's an exciting thought, as exciting as the idea that I can tell what kind of tea you like by feeling the bumps on your head. It is also exactly as wrong. After watching an hour or so of reality TV, you'd be forgiven for thinking that some people are only working at 10% brain capacity, but in actual reality, brain scans show that nearly every region of the brain lights up during even simple tasks, like walking and talking. Not only that, but the brain itself requires 20% of all the body's energy, and it would make little evolutionary sense to throw much energy away at something that's only minimally active. As animals, our capabilities have developed in part from our brain structures. We're actually able to trace our evolutionary history as we come to understand these structures. Less complex animals have simpler brains designed for basic functioning and survival, rest, breathe, eat, whereas more complex animals, like many mammals, possess brains that feel, remember, reason, and predict. These animals don't have all new systems. They have new brain systems built upon old brains. The brain is kind of like a set of Russian nesting dolls. The outermost wooden doll is the newest, most detailed, the most complex. As you go deeper, the dolls become older and smaller and simpler and more generic. The innermost wooden doll is the oldest, most basic, like a fossil in your head. This inner core of the brain, sometimes called the old brain, still performs for us much as it did for our early evolutionary ancestors. It's anchored by the brainstem, the most ancient and central core of the brain, where the spinal cord enters the skull. Above it, at the base of the skull, is the medulla. Here, old brain functions happen automatically without any conscious effort. The beating of hearts, the breathing of lungs, that sort of thing. The pons is perched on the medulla, and it helps coordinate movement. Above the pons, at the top of the brainstem, is the thalamus, a pair of egg-shaped structures that take in sensory information related to seeing, hearing, touching, and taste. The reticular formation is a finger-shaped nerve network inside the brainstem that's essential for arousal, which isn't necessarily what you feel upon seeing a particularly nice-looking human, but instead refers to things like sleeping and walking, pain perception, other important functions. The baseball-sized cerebellum, or little brain, swells from the bottom of the brainstem and is responsible for nonverbal memory, time, modulating emotions. It controls voluntary movement, like dance moves, but it also gets impaired easily under the influence of alcohol, hence the term tipsy. The old brain systems keep our body's basic functions running smoothly, the sort of stuff any animal might need. This is pretty much where the brain stops for reptiles. For higher functions, we look to the limbic system. This includes the amygdala, hypothalamus, and hippocampus, sort of a border region of the brain separating the old brain and the newer, higher cerebral areas. The amygdala consists of two line-of-sized clusters of neurons and is responsible for memory consolidation as well as both our greatest fear and hottest aggression. Stimulate one area of the amygdala and a docile family dog suddenly morphs into a bloodthirsty Cujo. Shift that electrode over just a tiny bit and that dog will be cowering at butterfly shadow puppets. The hypothalamus keeps your whole body steady, regulating body temperature, circadian rhythms, and hunger. Also helps govern the endocrine system, especially the pituitary gland. You should also thank your hypothalamus for allowing you to feel pleasure and reward. Rats implanted with electrodes in the reward center of their hypothalamuses and given ways to self-stimulate those areas will essentially reward themselves until they collapse or die. So use with caution. The final part of the limbic system is the hippocampus, central to learning and memory, and if it's damaged, a person may lose their ability to retain new facts and memories. Now, above all of this is the most advanced stuff, the stuff that you think of when you think of the brain, the gray matter. The two hemispheres of your cerebrum make up about 85% of your brain weight and oversee your ability to think, speak, and perceive. The left and right hemispheres govern and regulate differing functions, giving us a split brain connected by a structure called the corpus callosum. So, for instance, language production is controlled largely by the left hemisphere, while certain creative functions are controlled by the right. Well, this has nothing to do with handedness or people having dominant sides of their brain being more analytical or creative or whatever. That's part of what we call pop psychology, a behavioral disorder in which journalists and armchair psychologists use research showing beautiful, detailed, intimately connected complexities of your brain to sell newspapers or reinforce previously held beliefs. Yes, some tasks are distributed to one side, but the sides are deeply and constantly connected. A statement as general as artistic people use their right brains is as useless as saying artistic people have particularly bumpy heads. Finally, covering the left and right hemispheres, we have the cerebral cortex, a thin layer of over 20 billion interconnected neurons. But let's not forget the unsung heroes of your nervous system, the billions of non-neuron glial cells, which provide a spider web of support that surround, insulate, and nourish these cerebral neurons. You've probably seen enough brain diagrams to know that the cerebral cortex's left and right sides are subdivided into 4 lobes: the frontal, parietal, occipital, and temporal, all separated by especially prominent folds or fissures. Each lobe does indeed have its own set of duties that would have made Franz Gall proud. The frontal lobes, just behind your forehead, are involved in speaking, planning, judging, abstract thinking, and as the tale of Phineas Gage reminds us, aspects of personality. The parietal lobes receive and process your sense of touch and body position. At the back of your head, the occipital lobes receive information related to sight. And the temporal lobes, just above your ears, process sound, including speech comprehension. Remember that each hemisphere controls the opposite side of the body. So my left temporal lobe processes sounds heard through my right ear. And within these lobes, there are still more regions that have specialized functions. Your motor cortex at the rear of your frontal lobes, for example, controls voluntary movements and sends messages from the brain out to the body like, pet that dog, or pick up that mug. The sensory cortex right behind it processes incoming sensations like, ooh, that dog is soft, or God, that mug is hot. The rest of your gray matter is made up of association areas that are related to higher mental functions like remembering, thinking, learning, and speaking. But the thing about association areas is that unlike your sensory or motor cortex, you couldn't just poke one and create a response. Association areas are more subtle. They deal with things like interpreting and integrating sensory input and linking up with memories. And they prevail throughout all 4 lobes, so brain damage to different areas will cause very different results. A lesion on a specific part of the temporal lobe may destroy a person's ability to recognize faces. Traumatic memories or overactive hormones can profoundly affect our behavior and emotions, all of which remind us how fundamentally biology and psychology are intertwined. And there are a few more fascinating examples of this than how we sense and perceive the world around us, so that's where we're going to pick up next week. For now, if you were paying attention, you learned the basics of the central nervous system, specifically the brain, which can be understood in terms of older, more evolutionarily ancestral structures, along with the limbic system and new structures, which include lobes, cortices, and association areas. Thanks for watching this lesson in Crash Course Psychology, which was brought to you by The Air Show, Midnight House Elves on Etsy, and Daniel Vansky. Thank you so much to all of you. If you would like to sponsor an episode and give your own shout out, you can learn about that and other perks available at Subtle.com. This episode was written by Kathleen Yale, edited by Blake Pastino and myself, and our consultant is Dr. Ranjit Bhagwat. Our editor and director is Nicholas Jenkins. Michael Aranda is our sound designer, and our graphics team is Thought Cafe. Let me tell you about Oliver Sacks, a famous physician, professor, and author of unusual neurological case studies. We'll be looking at some of his fascinating research in future lessons, but for now, I just want to talk about Sacks himself. Although he possesses a brilliant and inquisitive mind, Dr. Sacks cannot do a simple thing that your average toddler can. He can't recognize his own face in the mirror. Sacks has a form of prosopagnosia, a neurological disorder that impairs a person's ability to perceive or recognize faces, also known as face blindness. Last week we talked about how brain function is localized, and this is another peculiarly excellent example of that. Sacks can recognize his coffee cup on the shelf, but he can't pick out his oldest friend from the crowd, because the specific sliver of his brain responsible for facial recognition is malfunctioning. There's nothing wrong with his vision. The sense is intact. The problem is with his perception, at least when it comes to recognizing faces. Prosopagnosia is a good example of how sensing and perceiving are connected, but different. Sensation is the bottom-up process by which our senses, like vision, hearing, and smell, receive and relay outside stimuli. Perception, on the other hand, is the top-down way that our brains organize and interpret that information and put it into context. So right now, at this very moment, you're probably receiving light from your screen through your eyes, which will send the data of that sensation to your brain. Perception, meanwhile, is your brain telling you that what you're seeing is a diagram explaining the difference between sensation and perception. Pretty meta. Now, your brain is interpreting that light as a talking person, whom your brain might additionally recognize as Hank. We are constantly bombarded by stimuli, even though we're only aware of what our own senses can pick up. Like, I can see and hear and feel and even smell this corky, but I can't hunt using sonar like a bat, or hear a mole tunneling underground like an owl, or see ultraviolet and infrared light like a mantis shrimp. Probably can't even smell half of what you can smell. No. We have different senses. Yeah. There's a lot to sense in the world, and not everybody needs to sense all the same stuff. So every animal has its limitations, which we can talk about more precisely if we define the absolute threshold of sensation, the minimum stimulation needed to register a particular stimulus 50% of the time. So if I play a tiny little beep in your ear, and you tell me that you hear it 50% of the times that I play it, that's your absolute threshold of sensation. We have to use a percentage because sometimes I'll play the beep and you'll hear it, and sometimes we won't, even though it's the exact same volume. Why? Because brains are complicated. Detecting a weak sensory signal like that beep in daily life isn't only about the strength of the stimulus. It's also about your psychological state, your alertness and expectations in the moment. This has to do with signal detection theory, a model for predicting how and when a person will detect a weak stimuli, partly based on context. Exhausted new parents might hear their baby's tiniest whimper, but not even register the bellow of a passing train. Their paranoid parent brains are so trained on their baby, it gives their senses a sort of boosted ability, but only in relation to the subject of their attention. Conversely, if you're experiencing constant stimulation, your senses will adjust in a process called sensory adaptation. It is the reason that I have to check and see if my wallet is there if it's in my right pocket, but if I move it to my left pocket, it feels like a big uncomfortable lump. It's also useful to be able to talk about our ability to detect the difference between two stimuli. I might go out at night and look up at the sky, and well, I know with my objective science brain that no two stars have the exact same brightness. And yeah, I can tell with my eyeballs that some stars are brighter than others, but other stars, they just look exactly the same to me. I can't tell the difference in their brightness. Are you done? Is it time for you to go? Give me a yeah. Okay. Good Good girl. The point at which one can tell the difference is the difference threshold. But it's not linear. Like, if a tiny star is just a tiny bit brighter than another tiny star, I can tell. But if a big star is that same tiny amount brighter than another big star, I won't be able to tell the difference. This is important enough that we gave the guy who discovered it a law. Weber's law says that we perceive differences on a logarithmic, not linear scale. It's not the amount of change, it's the percentage change that matters. All right. How about now we take a more in-depth look at how one of our most powerful senses works? Vision. Your ability to see your face in the mirror is the result of a long but lightning-quick sequence of events. Light bounces off your face and then off the mirror and then into your eyes, which take in all that varied energy and transforms it into neural messages that your brain processes and organizes into what you actually see. Your face. Or if you're looking elsewhere, maybe you see a coffee cup or a corgi or a scary clown holding a tiny cream pie. So how do we transform light waves into meaningful information? Well, let's start with the light itself. What we humans see as light is only a small fraction of the full spectrum of electromagnetic radiation that ranges from gamma to radio waves. Now, light has all kinds of fascinating characteristics that determine how we sense it. But for the purposes of this topic, we'll understand light as traveling in waves. The wave's wavelength and frequency determines their hue, and their amplitude determines their intensity or brightness. For instance, a short wave has a high frequency. Our eyes register short wavelengths with high frequencies as bluish colors, while we see long, low frequency wavelengths as reddish hues. The way we register the brightness of a color, the contrast between the orange of a sherbet and the orange of a construction cone, has to do with the intensity or amount of energy in a given light wave, which, as we just said, is determined by its amplitude. Greater amplitude means higher intensity, means brighter color. Someone just told me that sherbet isn't a word that exists. His name is Michael Aranda, and he's dumbhead. Did you type it into the dictionary? Type it into Google. Ask Google about sherbet. So sherbet is a thing. So after taking in this light through the cornea and the pupil, it hits the transparent disc behind the pupil, the lens, which focuses the light rays into specific images. And just as you'd expect the lens to do, it projects these images onto the retina, the inner surface of the eyeball that contains all the receptor cells that begin sensing that visual information. Now, your retinas don't receive a full image like a movie being projected onto a screen. It's more like a bunch of pixel points of light energy that millions of receptors translate into neural impulses and zip back into the brain. These retinal receptors are called rods and cones. Our rods detect grayscale and are used in our peripheral vision, as well as to avoid stubbing our toes in twilight conditions when we can't really see in color. Our cones detect fine detail and color. Concentrated near the retina's central focal point called the fovea, cones function only in well-lit conditions, allowing you to appreciate the intricacies of your grandma's china pattern or your uncle's sleeve tattoo. And the human eye is terrific at seeing color. Our difference threshold for colors is so exceptional that the average person can distinguish a million different hues. There's a good deal of ongoing research around exactly how our color vision works, but two theories help explain what we know. One model, called the young Hemholtz Trichromatic theory, suggests that the retina houses three specific color receptor cones that register red, green, and blue, and when stimulated together, their combined power allows the eye to register any color. Unless, of course, you're colorblind. About 1 in 50 people have some level of color vision deficiency. They're mostly dudes because the genetic defect is sex-linked. If you can't see the Crash Course logo pop out at you in this figure, it's likely that your red or green cones are missing or malfunctioning, which means that you have dichromatic instead of trichromatic vision and can't distinguish between shades of red and green. The other model for color vision, known as the opponent process theory, suggests that we see color through processes that actually work against each other. So some receptor cells might be stimulated by red, but inhibited by green, while others do the opposite, and those combinations allow us to register colors. So back to your eyeballs. When stimulated, the rods and cones trigger chemical changes that spark neural signals, which in turn activate the cells behind them, called bipolar cells, whose job it is to turn on the neighboring ganglion cells. The long axon tails of these ganglions braid together to form the ropey optic nerve, which is what carries the neural impulses from the eyeball to the brain. That visual information then slips through a chain of progressively complex levels as it travels from optic nerve to the thalamus and onto the brain's visual cortex. The visual cortex sits at the back of the brain in the occipital lobe, where the right cortex processes input from the left eye and vice versa. This cortex has specialized nerve cells called feature detectors that respond to specific features like shapes, angles, and movements. In other words, different parts of your visual cortex are responsible for identifying different aspects of things. A person who can't recognize human faces may have no trouble picking out their set of keys from a pile on the counter. That's because the brain's object perception occurs in a different place than its space perception. In the case of Dr. Sachs, his condition affects the region of the brain called the fusiform gyrus, which activates in response to seeing faces. Sachs's face blindness is congenital, but it may also be acquired through disease or injury to that same region of the brain. And some cells in the region may respond to just one type of stimulus, like posture or movement or facial expression, while other clusters of cells weave all that separate information together in an instant analysis of the situation. That cloud is frowning and running up into the tiny cream pie. I'm putting these factors together. Maybe I should get out of here. This ability to process and analyze many separate aspects of the situation at once is called parallel processing. In the case of visual processing, this means that the brain simultaneously works on making sense of form, depth, motion, and color. And this is where we enter the whole world of perception, which gets complicated quickly and can even get downright philosophical. So we'll be exploring that in depth next time, but for now, if you were paying attention, you learn the difference between sensation and perception, the different thresholds that limit our senses, and some of the neurology and biology and psychology of human vision. Thanks for watching this lesson with your eyeballs, and thanks to our generous co-sponsors who made this episode possible. Alberto Costa, Altna Agrawal, PhD, Frank Zegler, Philip Detmer, and Kurt Skazat. And if you'd like to sponsor an episode and get your own shout out, you can learn about that and other perks available to our Subbable subscribers. Just go to subbable.com/crash course. This episode was written by Kathleen Yale, edited by Blake DiDastino, and our consultant is Dr. Ranjit Bhagwan. Our director and editor is Nicholas Jenkins. The script supervisor is Michael Ronda, who is also our sound designer, and our graphics team is Pod Cafe. Homunculus. If you've heard that word, it probably wasn't in the context of psychology. A monster in Dungeons and Dragons, a song by the Movie Giants, the bad guys in Full Metal Alchemist, a novel by James Blaylock. It's Latin for little man, and in psychology, it refers to a kind of sensory map of the human body, a depiction of what we'd look like if each of our parts grew in proportion to how much we sense with them. Witness! Look at this dude. His ham hands could rip off a car door. I mean, like, if he could lift them, which he couldn't. What we're supposed to look like on the inside? This freaky thing illustrates the weighted significance of our sensory receptors. His disproportional hands are monstrous, for example, because we primarily touch the world with our hands, not our elbows, so our hands are extremely sensitive. That, meanwhile, is huge because we also have a ton of sensory receptors in our tongues and lips. It's what we use for tasting food, second face. As we continue our exploration of how we both sense and perceive the world, the homunculus, while kind of freaky, is actually a pretty attractive model for understanding how our bodies interact with the environment. How we smell it with our outside nose, taste it with our ridiculous Mick Jagger lips, and touch it with our enormous Donkey Kong hands. So join me as we get to know the hideous little creature within you. Last week, we talked about the difference between sensation and perception. How sensation is the process by which our senses and brain receive information from the outer world, while perception is how we organize and interpret that information and give it meaning. Like right now, my sense of hearing is letting me detect sounds, while my brain is processing and interpreting them, allowing me to identify and perceive the individual sounds and determine if they're coming from the radio, outside, or right behind me. Sound moves in waves that vibrate through a medium, like air. And although they function differently than waves of electromagnetic radiation, including what we think of as light, sound waves also can vary in shape. Short waves have a high frequency and a high pitch, like a plucky violin. Long waves have a low frequency and pitch, like a mournful cello. Wave height or amplitude determine a sound's loudness, which we typically measure in decibels. And just as light waves become electrical impulses that we register with our sight, so too do our ears turn vibrating air into signals that our brains can decipher. While the human ear might not be as elegant as the jackrabbit's or as wild as the long-eared bat, it's actually a pretty incredible organ. Or organs, since we have two of them, which helps give us directional stereophonic hearing. That's the 3D type of hearing that we couldn't experience if we had just one big freaky ear in the middle of our foreheads. Your outer ear, the part that you can see and pierce, tug on, collects sound waves, and funnels them through the ear canal into the middle ear, where they cause your eardrum to vibrate. From there, sound vibrations are amplified by the so-called ossicle bones, which also happen to be the most awesomely named bones in your body: the stirrup, the hammer, and the anvil. From here, those physical vibrations travel to the inner ear, where they bump into the snail-shaped cochlea, and its surrounding fluids get jostled around, causing some of your 16,000 tiny cochlear hair cells to bend. This motion triggers neighboring nerve cells that convert that physical energy into electrical impulses, zipping up the auditory nerve into the auditory cortex, where the brain is like, oh, songbirds or Elvis or vengeance, whatever. And it feels great with a little bit